most likely to be used when doing discovery work and samples from permafrost, i.e. moving beyond identification through DNA sequencing. It seems like at the moment the main way of doing it is doing DNA sequencing and identifying what are known to be pathogens already. And I suppose we've got a little bit stuck on that in the sense that there are other approaches you can do. So you can do lab-based culturing. But the trouble with that is that there's a lot of unknown diversity there. So you can't count on being able to culture it in the lab. Nonetheless, there is a need to improve the culturing techniques. But it is a big effort to do that anyway. I think we had a bit of, I wouldn't say we were fully in agreement about that, to be honest, but we did say that there is already a list of pathogens that we know. So the most efficient way of doing it is to sort of identify sequences from that list and use that to include or exclude pathogens. So we talked a little bit about using potential of things like proteomics to confirm pathogenicity, but again identified that is still quite a challenge and is perhaps something for sort of future directions. We don't really have perhaps a very good way of identifying them other than sequencing at the moment. Potentially metabolites that you could infer based on bioinformatic data, but that is just an indication. Which pose higher versus lower risks? There's quite an important distinction, I suppose, that we made between the environmental versus identifying pathogens in the environment versus in animal carcasses and that kind of thing. And I suppose there are indications from carcasses that you find, sometimes visual indications. But I think that the key thing that we decided was there needs to be a really strong procedure that's carried out there in terms of the DNA procedure, and specifically including making sure that a lot of metadata is included in that as well. In terms of the environment, I suppose it's very hard to kind of to have any indication about risk from the environment, but there are specific hotspots that there can be in the environment. We talked about a couple of specific examples of that, but it's very context dependent, but there can be environmental sites where there are clear environmental risks, and so that's something that needs to be taken into account. Level of risk for laboratories working on isolates from human and animal remains. BSL 2+, I think, was the term that was used, which I suppose is the level of risk that it would go to once you've identified that there's a pathogen in there. But you don't know that really until you've started the lab work and done that. I think that's about all I've got. Were there any other?
I think just one more thing or a few more things that were mentioned was kind of a moral obligation to preserve both the environmental and the clinical samples that are collected, and that's really difficult to do. There really are no resources to do it, but as our techniques improve in the future, it would behoove us to keep these samples for future generations. And kind of there is a struggle, a good struggle in a way, kind of the environmental microbiome versus the clinical and what techniques are used, and kind of as we progress from the environmental standpoint, that there's so much unknown that it would also be useful to start to culture these organisms, and we can use information from the metagenome assembled genomes to try to predict substrate utilization so we can do a better job of culturing these organisms. And I think just one more point that I'll mention is it was brought up of a systematic approach for sampling in these hotspot areas where a bunch of carcasses have been found, and trying to understand through, just kept on going back to DNA sequencing, kind of the dispersal of those organisms for predictive purposes. And thank you, David, for capturing everything for us.
So we had a difficult challenge, which was to, in the limited time we had, cover two of the research or the discussion topics. And I'll go through both 2 and 3 in turn. First, regarding biocontainment and engineering controls, which we took kind of in a broad biosafety and security framework. There was a lot of concern expressed about the fact that many people who may be working or visiting the Arctic may not be completely aware that the material they're working with could even contain pathogens. That could be a key awareness gap. And also an extremely limited amount of biosafety. Someone mentioned BSL 2+. Even that may be a real stretch in many parts of the Arctic. And then on the other hand, a lot of discussion about, well, given that context, what is the best way forward? Are we better served attempting to do top-down regulation, because after all, this is very serious and could be a real risk for humanity? Or on the other hand, would that sort of throw a wet blanket all over valid research questions, or B, potentially impede the building of capacity and the generating of community awareness by kind of coming in and imposing a lot of top-down regulatory control? Also, sort of an open question, we didn't dive into it very far, but while we'd like to think that everyone doing this research is doing so for benign reasons, that might not be a completely perfect assumption. There may be less than benign activities that need to at least be considered in any kind of discussion of this sort. Then there was a question about how to concentrate, I think, both the samples and more generally the data and information that comes from them. And a lot of opportunities, especially with new technologies and sort of a citizen science approach. Some questions about whether, kind of spreading those technologies out in a wide sense could perhaps complicate some of the biosafety and security concerns, but noted that this was not exclusive to this particular issue, that regarding synthetic biology and other things, we've had to deal with this issue in other venues. And then questions about if it isn't sort of a full-on regulatory treaty type instrument, are there other international frameworks or codes of ethics or best practice dissemination that we can use to make sure that affected communities are in the loop and that where relevant control of information and sovereignty concerns are appropriately addressed? And that might be highly specific to national context. And again, kind of a discussion about the role of bottom-up awareness raising and capacity building versus kind of a top-down regulatory approach, and some real trade-offs that have to be considered there. Moving to slide 3. Okay.
Oh, it's there.
It's up. Magic. I divined it. First off, regarding the question about which isolates and samples pose the greatest risks, was noted that we're not actually talking about the sample itself, it's the sample combined with whatever human activity leads to exposure, so it's an important point. A lot of different considerations that are kind of affecting our certainty here. We, by the way, asserted that we had very low certainty about what the risks were, and even a lot of challenges to achieving a higher level of certainty, much less a higher level of actual risk probability. So, real questions around whether a pathogen can survive in permafrost. It is a very hardy environment. It does take a certain something to make it there for very long. It's kind of the New York of the ecology world, I guess. Important questions about the probability of transmission. Just because something is surviving in permafrost doesn't necessarily mean it can be taken up by animals or humans. And then also a lot of questions about the severity of outcomes. So even something that might be fairly rare, but if it had some ability to, say, spark a global pandemic, that might be worth considering, even if it was a fairly low probability event. The magnitude of the impact is so high that it's worth considering. A lot of discussion about the trade-offs between animal versus human, and I guess you throw plant and animal samples. generally speaking, the thought was pathogens that are already known to affect humans. There's an important aspect to that. Not everything that affects other animals and plants affects us. But both given the statistic about the percentage of emerging infections coming from zoonotic sources and just the sheer matter, the biomass in the permafrost is largely animal and plant in origin and not that much human, it might change our equation a little bit in terms of what we're most worried about. We weren't even prepared to address the human-to-human transmission until we got some of these earlier issues sorted out. And then a really important discussion, can we scroll down this page? About doing risk assessments. And I think the bottom line was we need to know whose risk we are assessing and for what purpose. And the general sense was that we needed to start with occupational risk of people that might be involved in doing the work, as well as capacity building to do the work in the first place. Because if our sort of idea here is that the entry point is to start doing the foundational research, we need to make sure we're not accidentally infecting the researchers while we're doing that. So that was considered a kind of a foundational step. And then beyond that, there were a lot of questions about whether there was a need to focus on evolutionary history versus the present day as far as a risk assessment. The sort of subset of things that are both current pathogens and could conceivably survive in permafrost, whether there are certain key populations in humans or sentinel species in wildlife that are particularly important, and so on. But our bottom line was that if people are not doing this work in a safe way, that's a no-go, and we need to start with that sort of occupational risk as a first step. Anything I missed from the group?
Group 2 or 3? We missed? Any questions? Thank you.
So for group 4, we were dealing with the questions on impact on the communities. So the first questions was, are the communities living in areas near permafrost going to be exposed to animals, corpses and ancient pathogens regardless of whether scientific research is pursued? And we reached a consensus within 5 seconds that the answer is yes. But there are a number of qualifiers, and the first one is depends on what kind of permafrost is thawing. Some of it could be almost completely mineral, other it could be containing a high level of carbon, and so if it's largely mineral, then people are creating dust by traveling over these areas, and so to an extent there is an environmental exposure to it, from potential pathogens in the soil as the ground is opening up. So that particular exposure will take place, and it makes an important. You know, there are examples of course, that people are attracted to animal remains, so that, you know, there is a market developing in some areas for small mammals, and and Alexander gave the example that if you find a rhinoceros, you're well off these days. And so essentially, we can be absolutely sure that people because of their economic and social activity, are going to go and look at the material that is there, and to an extent that, of course, leads to the exposure. What is important is that, therefore, to an extent, these groups could be sentinels in terms of what might happen in terms of exposure to pathogens, and it was certainly we are at this moment not in a situation where we had many examples of that. Nevertheless, we were also discussing the fact that from the feces of caribou that had been frozen, people were able to come forward with a fungus that was able to infect plants. So it is possible. The earliest indicator that maybe, and it particularly relates to the fact of course that many human diseases are zoonotic, is that if animals live in the area and demonstrate particular disease symptoms, then that may be an early indicator of that something has happened. And I think there would be a lot of merit in trying to look at that. What was also clear is, and particularly if we talked about the fact that most of the communities have their own grave sites, it was quite clear that there are, in terms of exposure to human corpses, if people find a frozen corpse, then they go to the police or they try to make sure that there is some sort of follow-up. If it is human remains, then there are a large number of spiritual factors that come into account, in the sense that people feel that they should be, as a community, the first to decide what is happening now, because they look upon these as, obviously, as in most cases, their ancestors. And delicate situations can be generated as a result of leading to a situation which causes stress and also causes an unwillingness to cooperate with the researchers. So communities are worried about permafrost, and what is also quite clear is that essentially that communities are also going to be exposed because of this hunting for animal carcasses, which is a new business. Then the second question we had was, are there risks to food safety for the indigenous communities? And one of the main questions that was discussed was, has anyone developed best practice material to share with communities on food safety? And you need to be able to share that and don't have the time, but you don't have, there is an issue of resources and time. So the state of Alaska has a fact sheet on how to handle meats, but the problem, when we are talking, for example, about the potential to food safety, then there is also a food security issue if indeed a number of plant pathogens, for example, might come up. Although most of the areas don't have that much agriculture, of course. So
clarification.
Yeah.
So I'm sure the state of Alaska has a lot of fact sheets on how to handle meats.
Yeah.
But I think that point was actually supposed to slide down to the third question.
Okay.
And the point was that the state of Alaska has a fact sheet on how to handle human remains.
Okay. Right. Okay. Okay. So sorry, we got it. Thanks for that,
Cannibals.
And particularly in relation to that third question, of course, the problem was also that we were discussing what was going to happen in relation to the discovery of grave sites, was the fact that people don't know when they see corpses what they died of, and therefore to an extent that may make it difficult to assess to what sort of precautions they should take. Now, are there special precautions to be taken? Guidance is needed, obviously, for public health, for public officials, and for the communities. And I think that is an important element, particularly when it is relating to the handling of bodies of animals and humans that are uncovered from the permafrost. The main issue that came up here is that essentially it is very hard to see to what extent the people who are doing the hunting, the fishing and the camping are able to provide themselves with any form of protective equipment. It is simply not practical. the guidance that you could give, and that has been given apparently, is to say to people, if you see that there is a large area of thaw going on, don't go and camp there and try not to prepare your food there, and try to make sure that you handle that, avoid that area to some degree. But as I said, there are those providers, there is a proviso that people are attracted to those sites for other economical reasons. Then should the communities working with scientific organizations, and really the main organization that came through was of course the public health department, can be the first contact, but it is difficult in some areas to do that. The benefit of the public health authorities in this case, of course, is that they can communicate with the people by modern technology. Of course, there are practical difficulties in many cases for direct interaction between public health authorities and the people locally. And I think it was quite acceptable that one of the things that could be done in terms of the interaction with the authorities would be that if there are eroding grave sites, then that should be reported. But again, we get back to the question that people have in relation to the fact that they, of course, are the ones that want to determine in the first instance what is going to happen to the eroding grave sites, because it is their ancestors that we are talking about, and so spiritual factors are important. So I hope I have summarized most of what we discussed in a fairly lively session.
Yeah, thank you very much, and Katie for tremendous efforts. That's all I want to add, but we had quite a large group. If anybody else has a point of clarification or comment, please speak now.
I have a question from outside the group. Okay. It's related to an issue that we discussed in our group quite extensively, is when there is, the situation is to a certain extent reminiscent of what you do in normal diagnostics in a virological lab. Yeah, if you have indications that something strange is going on, for instance, if you were to have, like we have seen that in the past, if you were to have a graveyard where you have a lot of corpses from the 1918 flu, for instance, or similar things, if there are indications for poxvirus infections or mass mortalities or whatsoever, then you have an indication, then you upgrade your activities. If that's not the case, you are careful, and you can use it, you can analyze the stuff under the normal precautionary principles, but as soon as you find indications for one of those agents which are on the WHO list or on the BARDA list or whatever, then you upgrade your activities. But I think in the first instance it's very important to, for instance, if you see certain lesions, if that's the case, or you see a mass grave and you have indications for mass mortality or something strange that has happened, then you should be alerted, I think.
I think that was one of the discussions, because I posed a question in the beginning. As I said, are the indigenous communities afraid of approaching sites like that? And the answer that I got was, and please correct me if I'm wrong, was that if there had been a mass mortality, then they were. Yeah. There was that sort of reminiscence of that event. Okay.